Hi, everyone. So we're just going to give everybody a few minutes to be able to connect into the room. I know that many of you are the presenters here today. That's awesome. So excited. Heather and I had the opportunity to go through your posters. We are on the symposium committee, and we are so impressed with the quality of work that you all are doing. It's so exciting to be able to see the discoveries that you're making and what makes your environment unique. So bear with me just for a minute while I get everyone available. Wow, we got a lot joining in this room. So as a reminder, this is a safe environment, so no hurtful language will be tolerated in the room. If you have questions for the presenters, you can type your questions into the chat box. If we have enough time, we might be able to take some oral questions. You can raise your hand if we do have that time, and then we can unmute you and you'll have an opportunity to speak with the presenter. Each presenter has a total of 5 minutes. So if you can ideally, hopefully keep your presentation down to about 4 minutes with about 1 minute for questions. First up is going to be Jessica. On deck is going to be Madison. I am going to be giving you a 1 minute warning out loud. I know you have the timer up here, Heather. That's awesome. Thank you so much for putting that timer up there. But just as a reminder, you'll have 1 minute warning. We do have to get through several presentations today, so we want to make sure that we stay on time. So please try to keep your presentation right around that 5 minute mark. Jessica, are you here and available? Great. Hang on. I see you. Give me 1 second. I'm going to try to unmute you. Let me see here. Jessica, can we hear you? I cannot hear her. Hey, Heather.
Yep. Would we like to move on to the second one, and we'll let Jessica resolve the technical issues with the sound?
Okay, Madison, are you available?
Yes, I am available.
Okay,
Can you
awesome,
hear me? Okay.
You want to go ahead and introduce yourself? We'd love to hear about what you discovered.
Okay, so I'm Madison Marquis. I'm from UW Green Bay, presenting. I collected a soil sample from Voyager Park in Green Bay, Wisconsin. This soil sample was serially diluted by increments of 10 and then plated. 16 unique colonies did emerge. These were all tested for antibiotic activity against Staph epidermidis and E. coli. 6 colonies exhibited antibiotic activity, 3 of which showed broad spectrum activity. Further experimentation was carried out on these 6. PCR was performed in order to amplify the DNA. This information was then sent to the Yale Core facility for gene sequencing, and then blast analysis was carried out for confirmation of each isolate's genus. So further identification, 3 isolates were confirmed to be gram-negative based on gram staining, black as gram-negative, and all gram-negative isolates were did belong to the Pseudomonas genus. The other 3 isolates were gram-positive via gram stain, and blast analysis confirmed them to belong to the Bacillus genus. Further identification was performed via biochemical tests. All 6 were catalase positive. Thioglycolate broth revealed all 6 to be aerotolerant. The SIM test showed non-motility, no indole production, and no hydrogen sulfide production. There was some contradiction with the TTC media, which did show some to be motile, and so further testing would need to be carried out in order to kind of confirm those results. So starting with the gram-negative results, isolates 3 and 14 both appear to be Pseudomonas aeruginosa. Both patches exhibit blue pigmentation, which is characteristic of the species. Isolate 14 at first did not show this blue coloring, but isolate 3 did immediately. Isolate 13 was another gram-negative. This was also a member of the Pseudomonas. It's unclear which species. There were 3 which were non-hemolytic, which was what 13 was. However, all of those 3 were motile, so it's inconsistent with the TTC and SIM tests for isolate 13, which showed non-motility. So further experimentation would be required. Gram-negative result, or gram-positive, I'm sorry. Isolates 11 and 15 were most likely Bacillus cereus. These both showed similar beta hemolysis on blood agar plates, and both were microaerophilic. Isolate 10 seems most likely to be Bacillus subtilis. However, this did grow on McConkey agar, which is selective for gram-negative. So I am not exactly sure how isolate 10 grew on there. So further experimentation would also need to be carried out on isolate 10 to determine its identity. And those were my findings.
Awesome. Thank you, Madison. So if you have questions for Madison, you can go ahead and raise your hand and we can unmute you, or you can type them into the chat feature. So Madison, I have a question for you. So my courses, okay, here you have a question. How long did all of this take you?
This was carried out through the entire semester, so about 12 weeks, and each week we just did one part of each procedure of it.
So Madison, I had a quick question for you. So I am in Florida, and where I teach, our school is online. What was it like going into the classroom to have to do this type of work? Was it stressful?
It was a little stressful. So we did have, our labs are usually about 3 hours long, but it was divided so that in order to keep with social distancing, there were, I think, 8 students in our lab at a time. And then, so we only had an hour to do this. And so it was kind of difficult with the more isolates you had to get everything done within that time frame. But it did end up working out all right with our schedule. And we were happy to be able to continue going throughout this entire semester in person.
Awesome. Thank you so much, Madison. There's another question there. You can type back to Jess. She's asking approximately how large were your zones of inhibition? Did you take any measurements in millimeters by chance with a ruler?
Unfortunately, I did not measure them, so I would have to do this again to measure them out.
That's awesome, though. So great job, Madison. Thank you so much. Jessica,
Thank you.
Your microphone working?
Can you hear me?
Yes, great.
Okay, great.
So Jessica, if you want to go ahead and introduce yourself, tell us a little bit where you're from.
Okay, hi, my name is Jessica Forbes. I major in human biology at UW Green Bay. And I would just like to start by saying that this was all very eye-opening for me to learn about escape pathogens and antibiotic resistance. And it was very exciting to be involved in this research and to find antibiotics in soil that was very close to my home. I chose my sample site. I went to a public park in Appleton, Wisconsin. I've been to this park a lot of times, and there's nature trails there that I've walked, and I thought this would be a good place to take my sample from. The site I chose was abundant in plant life. This is a good indicator that the soil is rich in oxygen and nutrients, And it's also a good indicator that the bacteria in the soil has developed antibiotic qualities due to the high competition in the soil for that nice environment. When I went to the sample site, I dug down about 8 inches. I researched the best soil to find bacteria in, and it was loam soil, which is made up of sand, silt, and clay. My soil was dark brown and the granules were pretty large. Loam soil retains nutrients and water very well, therefore you can normally find a vast amount of microscopic life and therefore many different types of bacteria. Then a gram of the soil sample was serial diluted on Luria broth agar plates with containing cyclohexamide. Cyclohexamide was used to inhibit fungal growth. And the Luria broth plates were used because they're not traditionally used by scientists, so using them enables us to find and grow unique bacteria strains. The purpose of serial dilution was to derive a countable number of colonies. This number is between 30 and 300 colonies, and then we were able to calculate a colony forming unit per gram of soil. The individual unique colonies were picked and patched onto PDA master plates. I had 5 unique colonies emerge, 2 of which resisted E. coli and 2 resisted E. coli and 1 resisted Staph epidermidis. The isolated colonies were tested on escape pathogen safe relatives, which are what S. epidermidis and E. coli are. Gram positive and gram negative were used to determine broad spectrum versus narrow spectrum antibiotic activity. And the E. coli is a safe relative of K. pneumoniae, and the S. epidermidis is a safe relative of Staph aureus. And then the small amount of antibiotic colonies were mixed with forward and reverse primers to undergo a polymerase chain reaction. This amplified the 16S ribosomal RNA, which is a highly conserved RNA sequence. That's why we wanted to amplify it. And then Yale sequenced the amplified genetic DNA. And from that, I was able to identify one of my isolates as a genus of Pseudomonas. I did this using blast analysis. I was only able to do this with one because one colony of the sequence was inconclusive. And both of my unique colonies that emerged were both gram-negative, rod-shaped bacteria. And that's all.
Awesome. Thank you so much, Jessica. If you have questions for Jessica, please go ahead and type them in to the chat window. So Jessica, what are you planning to do with your isolates that you collected this semester? Are you planning to do anything with them next semester?
I don't think so. I believe we just got rid of everything that we used this semester.
Okay. All right, our next presenter. Thank you, Jessica.
You're welcome.
Bailey is our next presenter. Bailey, are you available?
Yeah.
Great. Go ahead, Bailey, introduce yourself. Tell us a little about yourself.
All right. I'm Bailey Summerfield, and I'm a senior at UW Green Bay. And so this semester I got a soil sample which I got from just a park across the street from campus. So it's right down next by the bay. I diluted that soil sample and we diluted that, and then I plated that onto LB agar with cyclohexamide so that we could jumpstart so we can inhibit eukaryotic translation and inhibit fungi growth. And then from there, it was picked and patched onto PDA and TSA plates which are minimal media to jumpstart antibiotic growth. And from there, then they were tested against strains of S. epidermidis and E. coli. S. epidermidis, which is a safe relative of S. aureus, and then E. coli, which is a safe relative of K. pneumoniae. And I only had one isolate that ended up having any zones of inhibition. And so the pictures that are shown are we did that twice, so it showed it twice, but it would only show inhibition when it grew on PDA plates. And then I amplified the DNA through PCR, and then we sent that over to the facility where they sequenced our DNA, and then I used that for blast analysis, of which I found the genus of my one isolate to be Pseudomonas. And then I did a gram stain on it, which showed that it was gram negative and Coxus, which like matches up with the majority of like Pseudomonas. And then over the course of 5 weeks, I isolated an ethyl acetate extraction from the isolate and tested that once again against S. epidermidis and S. epidermidis and E. coli, and it had a pretty large zone of inhibition against S. epidermidis, and then it was compared against the control of methanol. And then I underwent biochemical tests, which were a little funky because we started them the week before Thanksgiving break. So it was like 2 weeks before I was able to actually like see the results. But from what I saw, it was lactose fermenting and it was catalase positive and motile, but it was negative for every other reaction really. And yeah, that's it.
Awesome. Thank you, Bailey. Questions for Bailey can be typed into the chat window. So Bailey, what of the escape safes did you end up testing? So you tested against Staph epidermidis and E. coli?
Yeah.
Did you test against any of the other ones?
No, just those two. That way that we could have tested against a gram positive and a gram negative stain to see if it had broad spectrum inhibition or not. And it did when it was just the bacteria, but the ethyl acetate extraction only inhibited S. epidermidis.
Interesting. So when you were growing the bacteria up itself, you were able to inhibit both E. coli and staph, but when you did the chem extraction, you could only inhibit the staph?
Yeah.
Very interesting. Thank you, Bailey. All right, our next presenter is going to be McKenna. McKenna, are you available and with us?
Yes, I am.
McKenna, if you want to go ahead and introduce yourself.
All right, my name is McKenna Matuli. I'm from I go to UW Green Bay. So for my soil sample, I took it from Baird Creek Preservation Foundation here in Green Bay, Wisconsin. I thought that this would be an interesting place because of diverse plant life that it has. It has a lot of non-native species as well as native species growing right around the soil that I took. So from here, I did a serial dilution with water. I wanted to be able to find a readable plate where it wasn't too concentrated and I could count the colony forming units from those plates. Then from there, I picked and patched 20 unique isolates onto the PDA as well as 10% TSA agar. I did these two different types of agar because I wanted to see if it plated better on one versus the other. They both did have cyclohexamide on them as well to inhibit fungi growth on them so that my isolate would grow. Then on there, I tested antimicrobial activity versus the S. epidermidis as well as E. coli. And from there, I found 4 unique isolates that did show zones of inhibition against this. 2 of them were broad spectrum, isolate number 6 and isolate number 12. Both did show zones of inhibition against both of these safe relatives of the escape pathogens. But then 2 of them were narrow spectrum, isolate number 3 and isolate number 10. They only showed zones of inhibition against the S. epidermidis. Then I did a gram stain. They showed that all 4 of the isolates were gram-negative bacteria. And then I did a polymerase chain reaction as well as a blast analysis. The polymerase chain reaction did amplify the 16S rRNA gene, which was then able to be used as a fingerprint so that I could later find the genus of each individual isolate. After the PCR, I got enough material to then send to the Yale core facility so they could sequence the data. So the blast analysis did show the genus of each of the 4 isolates that I did find. Isolate number 3 was from the Leliota genus. Isolate number 6 was from the Pantoa genus. And then isolate number 10 and isolate number 12 were both from the Pseudomonas genus. These all had a very high match rate. And then I did a biochemical characterization that did kind of reiterate the same results that were for the genus that was showed by the blast analysis. Especially the catalase test showed that it was positive for all 4 of the isolates, which is indicative of the genus from each that they come from, as well as the obligate aerobe of the Pseudomonas genus, which was indicated in the blast analysis as well.
Questions? So we have a question from Jenny. I am not familiar with your strains that are not Pseudomonas. Are they known antibiotic producers? Did you find anything in the literature?
I did find that they are both known for antibiotic producers, but they are very rare. They're not very, obviously Pseudomonas is a more common one that we do find. But yeah, both of the isolate number 3 and isolate 6, the Pantoa and Leliota are known for that, but they are very rare. So this was a very interesting finding for this soil to find some rare bacteria that hopefully can go on for antibiotic discovery later.
Very interesting. It's always nice when you find new bacteria. What was the toughest or the trickiest part of the process?
I think just dealing with this with the COVID-19 measures that we did have to follow in lab. I think some of the procedures kind of had to be moved around as well as just a smaller class size to accommodate for distancing. I think this kind of did make it a little tricky to kind of get everything in that we needed and find the data. It also didn't leave much room to do many experiments over if we needed to, or kind of like a gram stain or things like that. So I think that definitely made it a little bit more tricky than if it would be done in past semesters.
Mm-hmm. Thank you. Any other questions? All right, thank you so much. Amanda, are you with us?
Yes, I am.
Great, Amanda. If you'd like to introduce yourself and share a little bit about what you discovered.
So my name is Amanda. I am a triple major at UW Green Bay, and I am in my last year of school. So I took my sample from literally my backyard in Oshkosh, Wisconsin. And as you can see, I had a little bit of a help taking my sample, Maggie, the Basset Hound. And from this wonderful sample that I collected. It was serially diluted into water to get countable colonies. As you can see, there's a wide variety of different types of colonies, and I believe I narrowed it down to 9 different types of bacteria. Only one of them, however, was an antibiotic producer. So I did a gram stain of it, and it was a gram-negative rod-shaped bacteria that tended to form in chains, which I thought was interesting. And then when we sent it for PCR testing, unfortunately, the results came back inconclusive. So, not really sure what it is. It came back with 4 or 5 different possible genus options, but the biochemical tests showed that it is indeed gram negative. It's motile, it does not produce indole, and it is anaerobic. So further testing would need to be done to determine what kind of bacteria it actually is. I did test it against the staph epidermidis and Escherichia coli to have an example against both a gram negative and a gram positive safe pathogen, and it was in fact effective against both. It was more effective against the E. coli than the staph epi, but they were both very effective. I would say that it would be very interesting to see if maybe with a better sample, if we could possibly produce a better result for the extractions. I think that might have been the biggest problem is I didn't have enough of a sample when the extraction aspect was done, so the extraction didn't turn out very well. So I think that would be kind of an interesting thing to do, but if there's any questions, I'd be happy to answer them.
Great questions for Amanda. You can please type them in the chat box. It's very interesting that your strain was anaerobic. How deep was the soil sample that you collected?
It was about 6 inches down from ground level, and it was taken from underneath an oak tree, actually.
Very cool. So were you able to do any PCR on your organism?
I did, but it came back inconclusive because I believe I only had 26 bases. So it came back with possible matches to a lot of different genuses. So it was too hard to try and determine which ones because there were like 6 or 7 different possibilities.
Any other questions? All right, thank you, Amanda.
Thank you.
Okay, Ashley et al., are you all available?
Yes, I'm here.
Okay, great. Are you presenting by yourself or are others presenting with you?
I'm presenting by myself.
Okay, great. Ashley, if you want to introduce yourself, tell us a little about where you're from and your classmates and what your discoveries you ended up making.
Hi, everyone. My name is Ashley Wilkins. I'm a senior at Cedar Crest College in Allentown, Pennsylvania. With the help of 2 of my peers, Mercedes Mello and Emily Bancini, both of which who have graduated at this point, I was able to isolate 2 different microbes that have antibiotic producing potential. AW119 was isolated this fall in 2020 from Cedar Creek Park, topsoil in Allentown, Pennsylvania. EB was isolated in 2018 from topsoil at a hotel in Ocean City, New Jersey. Both of these microbes were isolated on tryptic soy agar plates and screened against different pathogens, tester strains. AW119 showed inhibition against Bacillus cereus and Bacillus subtilis, while EB initially showed weak inhibition against Bacillus cereus and subtilis, as well as E. coli. We then did ethyl acetate extraction of EB, and from there we found that there was stronger inhibition. There was stronger inhibition against Bacillus cereus, Bacillus subtilis, as well as Pseudomonas fluorescens, and weak inhibition for Staph epidermis and E. coli. We also then were able to do some biochemical characterization tests as seen in the top table. Both AW119 and EB were both positive for catalase and were motile obligate aerobic microbes. We then did some Kirby-Bauer testing on them. AW119 was tested against 8 standard antibiotics, and we found that there were zones of inhibition for all of them except for bacitration. EB then underwent Kirby-Bauer testing as well against 14 standard antibiotics, and it was found to have zones of inhibition against all of them except for bacitration, inoside, and ethyl butyl. Additionally, we were able to send AW119 out for sequencing. We did this by amplifying the 16S rRNA gene using PCR and sequenced it to the family, the genus level. We found that it identified to the Sacrobacillus genus. The Sacrobacillus genus is a relatively new genus that has diverged from the Bacillus genus. What's interesting about this is that AW119 only showed inhibition against Bacillus cereus and subtilis, which are actually the two most closely related tester strains. We attempted to send EB out for sequencing as well. However, we did not have a high enough PCR product to do that. So we do hope to do that in the spring. Additionally, in the spring, we hope to do ethyl acetate extraction for AW119. As we saw with EB, initially, sometimes microbes will show weaker inhibition, and then the extract will show stronger inhibition. So for EB, we found with the extraction that it was more likely leaning towards being a broad spectrum antibiotic producer, while AW119 currently is only showing to be working against gram-positive bacteria. So we're hoping that when we do extraction that we may see some other inhibition against other tester strains. Thank you.
Awesome. Thank you, Ashley. Questions for Ashley? You can please type them into the chat box feature now. Great job, Ashley. Why do you think the Sacrobacillus micro was able to affect those close related strain of other bacilli?
Not really positive exactly the reason why. One hypothesis could be just the type of antibiotic that it's producing. If it was to naturally grow close to one of those other, genuses, it would be, much more effective to have an antibiotic that worked against a Sacrobacillus versus, Pseudomonas fluorescens. If it's not, very close in the environment, it might not be as effective to have an antibiotic that targets that. So it could just be based on where they grow. Honestly, though, I'm not entirely sure why.
Great. Other questions? Thank you, Ashley. All right, our next presenter, Chloe et al. Are you all here, Chloe?
Hi.
Chloe, are you presenting by yourself or are you presenting with others?
I'll be presenting by myself.
Okay, Chloe, if you want to introduce yourself, share a little about what you discovered.
Okay, my name is Chloe Oval, and I worked on this project with my peers Aditya Jo Kukar, Artis Banu, and Gabe Frist, and we're all a part of the Medical and Bioscience Research Program for Waukee Apex. Apex is affiliated with Waukee High School, and it just gives high school students the opportunity to explore career paths through various projects. And then obviously our project was Tiny Earth since we're here at this conference. And then through this project, we learn valuable technical and professional skills that we can take into future workplace environments. Anyways, we started this project back in late September when we first collected 3 of our soil samples on September 29th and then a 4th on October 26th. And then these were all collected around our facility, which is located in obviously Waukee, Iowa, and it's kind of around the Des Moines area. 2 of our soil samples were kind of collected near this trail leading to some soccer fields, and then the other 2 were collected near a wooded area next to our parking lot. And these all had a depth of about 3 centimeters. We then did our serial solutions, which were diluted with a 9:1 water to soil ratio, and those solutions were then plated onto our LB auger plates. And then once we had the growth from that, we used a sterilized toothpick to select individual colonies and patch them onto our master plates. And we let them in the incubator overnight, except the next morning, not only did we have our individual colonies growing, it looked like we also had a possible contaminant, which you can kind of see in figure 1 and figure 2 of the results. Even though that showed up, we were able to see that isolate C1 was resisting against the contaminating bacteria. So to test this again, we basically just plated the C1 bacteria 4 times onto a suspected contaminated plate, which you can see in the plate 2 or figure 2 right here. And we also just put an empty one just to confirm that it was contaminated, even though we highly suspected it was, and it was confirmed. However, we did have to redo our master plates. So we redid that just so we could go and screen further. So far, we have screened against E. carnivora, P. fetida, E. aerogenes, and A. bailey. And we used an OD 600 nanometer method for UV-Vis spectrophotometer to estimate the bacteria density for plating. And then so far, we've only had really 1 possible hit while screening, and that can be shown in figure A. Oops, that circle's supposed to be over isolate A4 right under it. We're not exactly sure if it was a hit per se, just because there are so few colonies, and we're not sure if it's actually has an inhibition zone or if it was just coincidental with how the colonies grew. So we're gonna have to replate that. We did run some PCR using 16S primers and gel green staining, which we can see in figure 4. Lane 7 represents our C1 bacteria, which the first time we ran did not work. And then lane 8 represents our contaminant bacteria, which worked pretty well. We don't have a picture on this because after we submitted this poster, we ran it again and we were actually able to get lane 7 to create a band where it was supposed to. And then all the other lanes are related to our morning class, so I won't be talking about those really. Our future steps include sending off our bacteria to probably Iowa State's lab so that we can get some DNA sequencing. As I mentioned before, we're planning to repeat testing for A4 against the bacteria colony against A. bailey, and then we also just we want to screen for more safe escapes in the future. And that's about it.
Awesome, thank you. Questions for Chloe? Well, thank you very much, Chloe. That's awesome. So it's going to conclude our poster presentations right now. Thank you all for attending. As a reminder, we're going to have our fun trivia coming up here at 3:15 Eastern time, 2:15 Central time zone. Hopefully you'll be able to make it for that session. Lucy will be hosting that. Heather, do you have anything you want to add?
Nope, sorry, I wasn't able to ask many questions while we were going, but great job, everyone. I was excited to see your research and everything that you were able to share with us today.
Yes, it's always exciting to see what you're finding in your backyard. Your backyard is drastically different than my backyard. I know that most of you are in snowy, cold environments. I am not. So I always like to see what you guys have to, the obstacles, the challenges you have to overcome working in the environments that you all are facing. So very exciting. Great job, everyone. That's going to conclude our session. We're going to have about a 15-minute break. And then if you would come back in the main room, connect in for the trivia. That link is posted on the Tiny Earth webpage.